I'm going to go over them again. But we're going to start here. We'll take a little break after we get done with this lecture. I think it's only about 30 slides. And then we'll start the epidemiology lecture. We're not going to get through that lecture, but we'll try to get at least halfway done with that lecture. Okay? Okay, so you guys should have some idea what we're talking about in the beginning here because you guys just did a full lab about recombinant DNA. So again, biotechnology, I'm not going to go over this too much, we already did it. It's the use of microorganisms, cells, or cell components to make a product. Specifically, recombinant DNA is basically taking genetic information that you want produced in some form and putting it into some other organism to help to make that organism produce whatever protein you want. Okay, vector. So we've talked about vector before. This is a different type of vector. We've talked about vector before in the sense of a like an arthropod, a bug that carries a disease as a vector. Here we're talking about a molecule of self-replicating DNA that is used to carry a desired gene to a new cell. Okay? A clone is a population of cells arising from this cell, each carrying a new gene. So a clone would be, you take a gene, you insert it into a vector, you put that vector into a single bacterial cell, and then you grow that bacterial cell into millions of bacterial cells that all have this gene. That population of cells would now be a clone of that original cell where you transform your DNA. So here's a typical procedure. So usually the vectors, when we talk about vectors, usually we're talking about plasmids, which are just these small circular self-replicating DNAs that are either man-made, or they can be isolated from bacteria. Typically the ones we use in lab are typically man-made because they have all the characteristics that we need. So you take one of these plasmids, and then you take a gene from any organism. Again, let's say you want to make insulin from human. So you take the human genome, you isolate the gene that makes insulin, you splice out that gene, and you paste it into this plasmid. And you take this plasmid with your insulin gene, put it back into bacteria, and now what the bacteria will do will replicate like bacteria does and grow into millions and millions of cells, and each one of these cells will be carrying this gene for insulin and will make insulin protein. And then you can harvest the insulin and make, therapeutics based on that. Okay, we went over the possible options that you could do with that. Okay, so again, we didn't go over this too much, but the process of selection is how you can ensure that your gene of choice got into the organism the way you wanted it to. So basically selection is the process of killing off all the bacteria that didn't get your gene and only keeping the ones that did. Okay, so that's selection. Mutation and site-directed mutagenesis. We talked about mutation. Site-directed mutagenesis is basically focusing mutation in a specific spot in the genome that you want to mutate. Again, site-directed basically meaning you direct it to a specific site in the genome. It's not just random. So restriction enzymes, you guys should be somewhat familiar with those now. And they were a big part of the lab we did on Wednesday. But they're basically enzymes that cuts very specific sequences of DNA. So as you guys are going through the lab, you see each restriction enzyme has its own specific, maybe 6, 8, 10 pattern of DNA sequences that it's able to cleave. So I'm not going to go over that video again. Here's just a few that you guys probably saw. Again, you can see they have very specific sequences that they cleave. So BAMH1 will only cleave GGATTC. It won't cleave anything else. Even if you make a single base pair substitution, it won't cleave it anymore. So it's very specific for the DNA sequence that it will cleave. So here's the basic strategy of how you clone a gene into a plasmid. So you take a plasmid, you cut it with a specific restriction enzyme. It will cut, assuming that your plasmid has the sequence that restriction enzyme recognizes, it will cut it in those places, and then that small piece of DNA in between the cleavage sites will fall out, basically leaving an opening. Now, if you take your gene and you ensure that your gene has those same restriction sites, it'll basically just paste in nicely into that little opening and reclose the circle. Okay, so again, that's something that you guys did in the lab the other day. So again, you cut with a restriction site, make an opening, and then you basically paste your gene into that opening. And now you have a plasmid that has your gene in it. So here's a specific plasmid. You guys were working, I can't remember the name of the one you guys were working with in the lab. but it was similar to this. So the three important things about a plasmid, a functional plasmid, is that it has some kind of resistance gene that you can use for selection, and we'll talk about that in a minute. It has to have restriction sites because you need to be able to cut it with a restriction enzyme to open it up to allow your gene to be pasted in. It has to have an origin of replication, so it has to be able to be replicated. And what's also helpful in many cases is you have a reporter gene, and we'll talk about each of these in a minute. Okay, so you've isolated your gene, you know you want to paste it into a plasmid, and you want to get that plasmid into a cell. Okay. But when you isolate your gene, typically you only have a single copy or a handful of copies of that gene coming from the genome. You want to be able to replicate it and make millions of copies of that gene, so you can paste it into a million different plasmids. Okay? And the way you do that is by PCR. We talked about PCR on Wednesday. So typically, what we talk about, we're using it to amplify DNA, so to make multiple replicates, identical replicates of the gene. So this is basically how it works. And again, I went through this on Wednesday. So here's your gene. You've isolated your gene. You want to paste this gene into a plasmid, but before you do that, you need to make millions and millions of copies of this gene. Okay. So here's your gene. You basically incubate it at a very high temperature. That causes the double-stranded DNA to break into two single strands. Okay. You add these chemicals, basically all the bases that are needed to build new DNA, DNA polymerase, which is needed to replicate the DNA. You put them all together in a tube, and you incubate them at 60 degrees. That allows these little DNA sequences called primers to bind. What the primers do, it basically just allows a place for DNA polymerase to dock to the DNA. Okay, so the primers are now bonded, and then at 72 degrees, the DNA polymerase will bind to these primers and start to add the sequences to make these single-stranded DNAs now each double-stranded. So you've gone from a single double-stranded DNA to two double-stranded DNAs. If you do this whole cycle again, now these two double-stranded DNAs will turn into 4 will turn into 8 will turn into 16, so forth and so on. So every round of this temperature changing doubles the amount of DNA in the tube. Okay. So again, you just repeat this, and after just a handful of cycles, you've turned a single copy of that gene into a million copies. Okay. So now you've been able to amplify your gene, you've pasted it into a plasmid. Now you need to figure out, how do I get this plasmid into the bacteria? Okay. So there's a couple of different ways you can do that. Protoplast fusion is not typically what you would do. Protoplast fusion is a way to get DNA from one cell into another, but it's very non-specific. It's basically if you wanted the whole genome of one cell to move into another cell, you would use protoplast fusion. It's basically just forcing the two cells to merge together to form a single cell. Okay, so this cannot be used to insert a specific gene. So when we talk about transformation, in order to get a gene into a cell, protoplast fusion is not what you would use. What you could use would be any of these 4 techniques. Okay, microinjection. I have gene gun and microinjection are very similar. It's basically taking a very fine needle and puncturing the cell and injecting the DNA that way. Electroporation is basically taking cells and putting a strong electric shock into the cells that causes small openings in the cell wall, and then you mix that with the DNA goes into the cell, and then the cell repairs its cell walls, but the DNA has already gotten into the cells. Transformation is very similar to electroporation, but instead of causing these small holes in the cell wall with electricity, you do it with chemicals. So you add something like a high amount of calcium chloride, basically causes all the channels in the cell, all the transmembrane channels to open up and allows the DNA to get into the cells. So most of you guys have probably seen some kind of picture about this. Like artificial insemination is typically where you see this, where they take an egg and they inject DNA into it in order to hopefully get fertilization. So again, here's your very fine needle injecting DNA directly into the cell. All right, so let's skip over libraries. We'll go to selecting a clone, okay? So now you have your plasmid, you've cut your plasmid with restriction sites, you've pasted your gene into the plasmid, and now you've gotten it into the cells using one of those techniques we just talked about. But now you need to check and ensure that the DNA actually got into the cells. Up to this point, you have no way of knowing for sure that any of these processes actually work. Okay? You're just basically relying on the protocols. Okay? But that's where all these other parts of the plasmid come in. The resistance gene, the reporter gene are all very important here. So basically what you have is usually a resistance gene to some kind of antibiotic. Okay? And what that helps you do is you take the cells that you've transformed with your plasmid and you grow them in media that has, in this case, ampicillin in it, which kills bacteria. Okay? Any bacteria that got this plasmid will have the ampicillin resistance gene. Okay? So it will survive an ampicillin. Okay? Any bacteria that did not get your plasmid will be killed by the ampicillin. Okay? So if you grow your cells in media with ampicillin for a day or two, any cells that survive, you can be ensured has your plasmid in it. So it's a way of killing off all the cells that didn't take in your plasmid. When you do the transformation or electroporation, the rate of success of getting the DNA you want into the cells is about 5 to 10%. So you have about 90% of the cells in your culture didn't take up your DNA that you wanted it to. And you need a way to kill those cells off so that you have a pure culture of cells that only have your DNA. So again, the way you do that is you grow them up in a media that has some antibiotic in it that will kill all the cells except the cells that have your plasmid. Okay, does everybody see that? Okay, so now those 5 or 10% cells that took up your DNA, those are the only cells that survived. So now you can continue working with those cells knowing that they have your plasmid in them. Okay? Now, you know all the cells have your plasmid, but what you don't know is that all the plasmids actually incorporated your gene. Okay? Because possibly when you cut your plasmid, there's always the possibility that your plasmid just seals back up on itself. And the gene you tried to get into your plasmid didn't actually get into the plasmid. So you need a way to ensure that your gene actually got pasted correctly into your plasmid. And that's where the reporter gene comes in. Okay? And typically what the reporter gene is when that gene is intact, so here's a plasmid that doesn't have any foreign DNA in it. So let's say this plasmid got cut at the restriction site, and instead of getting, so here's that little orange would be your gene, instead of getting the foreign DNA, the gene, pasted into it, it just pasted back on itself. Okay, so it didn't get your gene. So it has an intact reporter gene. In this case, it's a LAC-Z gene. Okay, the plasmids that got your gene of interest pasted in basically interrupts this reporter gene. So this LAC-Z gene, this red, gets interrupted by your gene. And so there's not a functional LAC-Z gene in that plasmid. Okay? Here there's a functional LAC-Z gene because there's nothing interrupting it. Here your gene gets in the way, so there's no functional LAC-Z gene. Okay? And what you do, you have to grow these cells on a media that allow this gene to report out whether or not there's an intact gene. So in this case, you grow it in media that has a chemical called XGAL. Okay? If the cells turn blue on XGAL, it means they have this beta-galactosidase gene. Okay? If they turn white, it means they don't have beta-galactosidase. Okay? So anything that the plasmid pasted back on itself without your gene will have an intact LAC-Z gene. It'll produce beta-galactosidase, and when it's grown in the presence of XGAL, it'll make the cells turn blue. Okay? Any cells that have the plasmid with an interrupted LAC-Z gene will not make beta-galactosidase, again, because your gene is interrupting that gene. And without beta-galactosidase, there's nothing to make the cells turn blue, and they remain white. Okay? So at this point, because there's ampicillin on this media, the only cells that grow at all are cells that have your plasmid. And the only cells that are white are cells that have your plasmid, in which the plasmid also has your gene of interest in it. If they're blue, they have the plasmid, but they don't have your gene of interest in the plasmid. Okay. It's just an empty plasmid without your gene. Okay, so that's how you select a clone. So basically, you get to this point, you select all the white cells, and these white cells will have the plasmid, and that plasmid will have the gene of interest in that plasmid. Okay, so that's the process of selection. Okay, so depending on what you want to do with that gene of interest, you would grow it in various types of cells. Okay, so here there's some options on how you might want to, what type of cell you'd want to transform your plasmid into. So E. coli is a very common one. It's used because it's easy to grow, so it grows very quickly. A lot is known about its genomics. It typically doesn't have any endotoxin, so any products that are made from it, you don't have to worry about there being toxins in it. The negative is that the cells must be lysed to get the product. So the product stays within the cell, and to get the product out, again, whatever protein is from your gene of interest, you have to actually lyse the cells. A couple other options: yeast, Saccharomyces cerevisiae. It's also easily grown. It's usually for eukaryotic genes. So genes from eukaryotes you typically grow in yeast. Genes from prokaryotes you typically grow in E. coli. You can use plant cells. They're not quite as easy to grow as yeast or bacteria. And sometimes, depending on how complex the gene is, you may have to actually use mammalian cells, which are much harder to grow. So why would you do this? Why would anybody do this whole recombinant DNA technology? So sometimes, again, to make human enzymes and other proteins, so again, for instance, insulin. So in order to make large amounts of insulin that can be used as a medical product, we isolate the insulin gene from human, we put it in a plasmid, we put that plasmid into yeast, and we grow that yeast up, and then we harvest the insulin that's made from the insulin gene. Some subunit vaccines, so these are vaccines that actually contain proteins of the pathogens that we're trying to vaccinate against. So we use it in that case. DNA vaccines, gene therapy, these are all possibilities where we use recombinant DNA. There's also some scientific applications of looking at DNA from a pathology point of view. So if you look at DNA from bacteria or a virus, you can usually use that to identify where an infection came from. Okay, so for instance, here's an example. So let's say in this specific situation, there was an E. coli outbreak, E. coli food poisoning outbreak. And they didn't know where it came from. So they knew everybody that got this outbreak ate at a specific restaurant. So they took food samples from different types of food at this restaurant and looked to see if there was E. coli contamination by looking at the DNA. Okay, so again, each organism has its very specific DNA pattern. Okay, so what they found was that they found in the apple juice there was some E. coli, okay, based on the DNA process. They looked at E. coli infections from other people that didn't eat at the restaurant, so just a variety of E. coli infections. You could say each one is slightly different because each one's from a slightly different strain of E. coli. No two strains of E. coli are exactly the same. But when they look at the people who actually got food poisoning from eating at this restaurant, they noticed they were all identical to the apple juice E. coli. So this is how they could deduce that the people got food poisoning from drinking the apple juice at this restaurant. Okay, another example, a norovirus outbreak. So a virus, this was a bacteria situation, here's a virus situation. So here's a real example where they had a norovirus outbreak that started with a school meal. So there was viral contamination in some of the food that was served at a school. So you can see on day 2, that's when the meal was served. On day 3, there was just a couple cases of school employees. Okay. A couple days later, or a day later, there were more school employees. There were now other people in the community, publishing company employees. There was social service, or there were other reported community cases. And as you see, as days went on, it started to spread a little bit from the initial source. But again, they used DNA analysis to be able to trace it back, to figure out where it came from. And so they didn't get to it until it was, at day 6 and a bunch of different groups had it. What they were able to do is look at the DNA and trace it back and isolate it to the DNA from the norovirus that was found in the original school meal, and that's what they were able to figure out where it came from originally. Okay, so here's a very common application of using genetics to determine the pathogenicity of a disease or an infection. Okay, so that's it for that lecture. Let's go, we'll take till 6:15, and then we'll come back and we'll start the epidemiology lecture. Okay.